Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about permeabilization principle and why is it important and the protocol of the permeabilization. So as you know that the permeabilization is one of the most important part of the immunofluorescence staining and ISC, immunohistochemistry. So let's begin. First we're going to talk about the principle. So as you know that the cell has the cell membrane, intact cell membrane, and this cell membrane is composed of the hydrophilic and hydrophobic layer. So this hydrophobic layer contains the phospholipid, that is the lipid layer. So because of this kind of intact orientation of the hydrophilic and hydrophobic layer, any kind of macromolecule, for example antibody, it cannot get enter into the cell. So in the cell, on the cell surface there are some kind of proteins that present. Those are the localized protein on the cell surface. So without the permeabilization, this kind of proteins can be targeted by the antibody. So there is no problem in this case because this kind of localized protein, these are present on the cell surface. But if we target any kind of protein that localized inside the cell, in that case we cannot use this antibody without the permeabilization step. So here the permeabilization, this concept is arised. So in case of this kind of intact cell membrane, this kind of antibody, it cannot get enter and they cannot bind with the target protein. So this is why we use permeabilization. So permeabilization step, it removes more cellular membrane lipids to allow large molecules to get inside the cells. So we usually used the permeabilizing agent Triton X100 and Twin 20. So usually we commonly used the Triton X100 in our lab. So these are the organic solvent, and this organic solvent, it dissolves the part of the lipid layer of the cell membrane. As a result, the pores are created on the cell membrane, and this kind of pore, it allows the macromolecules to get inside the cells. So as a result, the antibody the antibody that we gonna use for targeting the protein, so this antibody, it can easily get inside the cells and it bind with the target protein. So this is why these permeabilization steps are very much important, and this step is very much important for the immunohistochemistry and IF staining. So now talk about the protocol of this permeabilization step. So usually for the cell we use normally Triton X100 1% for 5 to 10 minutes at the room temperature, and for tissue we use also the Triton X100 1% for 30 minutes at the room temperature. But the percentage of the Triton X100 and also the time, the incubation time, it also depends and it can be varied, and sometimes you need to be optimize the protocol. So after using this Triton X, you need to wash the cell or the tissue twice or thrice with the PBS, and then you need to continue the rest of the staining steps you need to do. So after proper permeabilization step, we can get the accurate expected result. So remember that without the permeabilization, this antibody, as I mentioned, that this antibody cannot get inside, and this kind of antibody, it cannot bind with the target protein. So this is why you cannot get the proper and accurate result without the permeabilization step. But after proper permeabilization, we can get the accurate and expected results. So this is all about the permeabilization principle, protocol, and the significance. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and if you have any queries, kindly write in the comment section. Thanks for watching.